Okay, let's unpack this. You've brought us a truly fascinating stack of sources.
Yeah, definitely.
We're diving deep into the world of computational modeling, specifically how computers are becoming, well, indispensable partners in figuring out what happens to a drug inside the human body.
That's right. This isn't just about finding a molecule that hits a target. It's about everything that happens after you take it, how it gets in, where it goes, how it changes, and how it leaves.
Right, the whole journey.
Scientists call this drug disposition,
Mm-hmm.
and you often hear the acronym ADMET.
Absorption, distribution, metabolism, excretion, and toxicity.
Got it.
Exactly. And your sources make a really powerful case for why this modeling is so crucial now. Historically, the focus was heavily on just like that initial target interaction.
Totally. But the reality is half of potential drugs actually fail in later clinical trials.
Half. That's huge.
Yeah, and often not because they don't work on the target, but because of problematic ADMET properties.
Think of the time, money, and, well, resources lost there. It's staggering.
Exactly. It's a massive hurdle. And that's the core problem computational modeling aims to solve.
Okay.
Predicting these complex ADMET characteristics before a drug candidate even gets synthesized or tested in the lab.
So moving the prediction way upstream.
Precisely.
So our mission with this deep dive is to take this stack of material and give you a shortcut, a clear picture, hopefully, how computational modeling has transformed pharmaceutical sciences, leveraging, increasing computer power and smart algorithms to really understand and predict how drugs behave.
It offers a more quantitative and mechanistic way to see that journey through the body under different conditions. It's not just guesswork.
Right.
We'll explore the different types of models used, trace a drug's path through the ADME stages.
Highlighting the specific computational tools for each step.
Yeah, exactly. And we'll touch on the current challenges and look at where this field is heading.
Ready to get well informed on how silicon and biology are teaming up to speed up the journey from a chemical idea to a safe and effective medicine?
Let's do it.
Let's dive in. All right, so the big picture starts with understanding drug disposition itself. As you said, it's the movement of the drug molecule through your body after you take it. It's that critical ADMET sequence.
And the core challenge your sources emphasize is that historical blind spot, focusing just on efficacy. Well, it meant drug candidates would get all the way to expensive clinical trials.
Only to fail then.
Right. Before discovering they were poorly absorbed or got stuck in the wrong tissues, metabolized too quickly, or were just plain toxic.
Ouch! That's the bottleneck. The in silico approach, computational modeling, directly addresses then.
It does. By building virtual models, you can screen molecules for potential ADMET issues early, way early.
Saving immense effort downstream.
The impact is huge. You reduce the need for animal testing, which has, ethical considerations and cost implications.
Right.
It gives researchers and industry powerful tools to predict how a drug's concentration will change over time, to understand why individuals might respond differently.
Based on things like genetics.
Yeah. Or environmental factors.
Yeah.
And even predict how different drugs might interact with each other inside the body. DDI, drug interactions.
And all of this is really accelerating thanks to the explosion in computational power, isn't it?
Oh, absolutely. We can run incredibly complex simulations now that weren't possible even a decade ago.
And the modeling algorithms themselves are getting much smarter.
They are. That's why you see so many different software tools emerging in this space.
Okay, let's look at those tools then. Your sources lay out a diverse toolbox of modeling techniques, each suited for different questions and levels of detail.
Right. On one end, you have the classic approach. Compartmental models.
Okay.
Think of these like a simplified diagram of the body, maybe just a few boxes.
Like bloodstream is one box, everything else is another.
Sort of, yeah. Central compartment for blood, maybe a peripheral one for tissues. They describe how a drug moves between these boxes using mathematical rates.
Like how fast it's absorbed or eliminated?
Exactly. You fit these rates, these parameters like Ka or K, to experimental data. Software like Phoenix Winnolan uses this approach.
So they're good for describing what you see in the data.
Yes, but they often lack physiological interpretability. They don't really tell you why it's happening from a biological standpoint.
Which brings us to a much more complex and arguably more insightful approach. Physiologically based, pharmacokinetic or PBPK models?
Yes, PBPK. These models build a virtual body. It's much more mechanistic.
Representing actual organs and tissues as interconnected compartments.
That's it. And they fill these virtual organs with real biological data. Things like the actual volume of the liver or kidney, the rate of blood flow through them,
Even enzyme levels.
and transporter expression levels in those tissues, yeah. Each compartment has parameters based on real-world physiology, not just fitting data points.
Okay, so instead of just a generic peripheral compartment, you have a virtual liver, virtual kidney, virtual brain.
Precisely. And this allows you to do some incredibly powerful things.
Like simulating drug behavior in specific populations.
Exactly. Say someone with impaired kidney function, or a child or an elderly person. You just change the relevant physiological parameters in the model.
Wow. So you can predict concentrations in specific tissues, too. That sounds critical for safety.
It is, and for efficacy. PBPK models are now pretty standard practice for optimizing doses, assessing safety. They're increasingly accepted by regulatory agencies, too.
Useful for extrapolating across species as well.
Yeah, and disease states. Major platforms here are SimSip, GastroPlus, and there's an open source one, PKSim.
Okay. Beyond those detailed mechanistic models, you have approaches that look for patterns connecting a molecule structure to its properties or activity. QSIR and QSPR models.
Right. Quantitative structure activity or structure-property relationships.
So these statistics.
Statistics, and often tapping into machine learning now to find correlations. They ask, does a molecule's size, how fatty it is, its lipophilicity or its charge predict, say how permeable it is or how toxic it might be?
Sounds great for screening lots of compounds quickly.
They're fantastic for that. Virtual screening, early filtering.
And speaking of machine learning, AI and ML are rapidly advancing this field, aren't they?
Hugely. Using techniques like deep learning or random forests, these models can analyze vast amounts of data.
And find complex relationships.
Yeah, non-obvious patterns between a drug structure or other factors and its ADMET properties, like predicting transporter interactions or toxicity risks.
They handle lots of data well.
They excel at high-dimensional data and uncovering those complex nonlinear relationships, perfect for the messy reality of biology. They can directly predict properties like solubility or plasma protein binding just from its chemical structure, using what we call descriptors.
Mathematical fingerprints of the molecule?
Sort of, yeah. Representations of its features.
And your sources mention that combining these approaches is often most effective.
Definitely. Like using an ML model to predict a specific parameter, maybe transporter affinity.
And then feeding that prediction into a PBPK model.
Exactly. For a whole body simulation. That creates really powerful hybrid models.
What tools are used for ML here?
Things like ADMET Predictor, DeepShem, and various libraries in Python and R are very active in this space.
Are there other model types we should know about?
Well, there's population pharmacokinetics, or POPPK, that analyzes data from groups of patients.
To understand variability.
Right. Quantify variability in drug response and identify influencing factors, covariates like age, weight, or genetics. This often uses specialized statistical software like non-mem or Monolix.
Okay. And simpler methods?
Yeah, non-compartmental analysis, NCA. It's model independent.
Meaning?
It just estimates key PK parameters like total exposure, AUC, or clearance directly from the measured drug concentration data over time, often used for initial data crunching. Phoenix Winterland does this too.
Got it.
And then at the most granular level, you have molecular modeling and simulation techniques.
Docking,
molecular dynamics, yeah. They peer into the atomic world, simulating how a drug molecule actually interacts with a specific protein like a metabolizing enzyme or a transporter.
Crucial for understanding the mechanism behind ADMET.
Absolutely. So yeah, it's not just one type of modeling, it's a whole suite of approaches, often used together depending on the question you're trying to answer.
And key software platforms mentioned across the sources are GastroPlus, SimSip, PKSim, EDM Predictor. Quite a few.
It's a rapidly evolving field.
Okay, let's follow a drug through the body and see how these models are applied at each stage of ADME, starting with absorption.
Right, that critical first step, getting the drug from where you took it, usually your gut for a pill, into your bloodstream.
And the biggest hurdle here, highlighted by your sources, is often solubility, right?
Absolutely. A drug has to be dissolved in fluid to pass through membranes. If it doesn't dissolve well,
you won't absorb much.
Exactly. You get low or unpredictable effects. It's a major reason drug candidates fail.
The Biopharmaceutics Classification System, BCS, categorizes drugs partly based on solubility and permeability.
Yes, and classes 2 and 4, the poorly soluble ones, are particularly tricky.
And it's striking that something like 40 to 70% of new potential drugs fall into these challenging categories.
It really is. So predicting that solubility before you even make the compound is incredibly valuable.
Using QSPR models.
QSPR models are common here, yeah. correlating the molecule structure with its predicted solubility, using those descriptors we talked about.
Molecular weight, hydrogen bonds, lipophilicity, like log P.
Log P is very important, yes. It relates directly to how easily a drug might move through fatty membranes. There are different ways to calculate it, ALOGP, CLOGP. They're widely used.
And newer MLAI models are getting good at this too.
Very good, especially deep learning ones that can learn complex patterns directly from a drug's structure. They are fantastic for high-throughput screening.
Predicting solubility for millions of compounds quickly.
That kind of scale, yes.
It's also not just solubility in pure water that matters.
No, the sources emphasize predicting solubility in biorelevant fluids, things that mimic your actual gut contents, like simulated intestinal fluids.
IFIF, FESIF, those kinds of things.
Right. Models incorporating those details are much more predictive of real-world absorption.
And tools like GastroPlus have sophisticated absorption models.
Yes, GastroPlus uses its ACAT model, simulating the gut environment, regional pH, bile salts, particle size impacts. ADMate Predictor also predicts various solubility types, log P, P-Shay.
PDA tells you if the drug is charged.
Yeah, at different pH levels, which affects solubility and permeation. And SwissADN is a handy free web tool for a quick check on many properties.
The case studies really bring this to life. Roche using GastroPlus for R1350.
Right, a poorly soluble drug. The model correctly predicted its solubility wasn't the main issue, and that shrinking particles wouldn't help absorption much.
Saved them going down the wrong path.
It did. And for aprepitant, a BCS Class 2 drug, the GastroPlus model accurately predicted the clinical finding that food significantly increased absorption.
Because of bile helping it dissolve.
Likely, yes. The model could then be used to simulate nanoparticle formulations to try and reduce that food effect.
Okay, so assuming it dissolves, the drug then needs to permeate across the intestinal lining, another key part of absorption.
Right. And computational models, again, QSIR and ML primarily, predict how easily a drug can cross those cell membranes.
Trained on lab data, like CACO2 cells.
Often, yeah. Or artificial membranes. They look at descriptors like size, lipophilicity, polar surface area.
But these models have limitations, especially with active transporters.
They do. Transporters, those protein pumps and channels are absolutely crucial. They can pull drugs in or push them out.
Affecting absorption, distribution, excretion, everything.
Pretty much. And PBPK models become really powerful here because they can explicitly incorporate the activity of these transporters in the gut wall.
So models in SimSip or GastroPlus simulate transporters working with dissolution and passive permeation.
Exactly. It gives a much more complete picture of how much drug actually gets absorbed.
And at the really fine-grained level, molecular dynamics.
MD simulations can give atomic scale insights into how a drug molecule actually wiggles its way through a lipid membrane passively, calculating the energy needed.
Wow, okay, beyond solubility and permeability, absorption models also account for dissolution.
How fast the drug dissolves, yeah.
Yeah.
And GI transit, how quickly it moves through the gut.
And first-pass metabolism. Metabolism before it hits the main bloodstream.
Right, in the gut wall and liver. PBPK models can simulate all these factors. They incorporate enzyme levels, transporter activity.
Even how food changes things like stomach emptying.
Yes. Predicting complex effects, like how different formulations or food might impact the total amount absorbed.
Okay, so once a drug is absorbed into the circulation, it begins to distribute throughout the body.
Its journey from the bloodstream into various tissues and organs, yeah.
How far and where it goes depends on, well, several things.
Plasma protein binding, PPB, tissue permeability, blood flow to tissues, and the drug's own properties, lipophilicity, charge.
Quantified by VINA, volume of distribution, or KP, tissue to plasma partition coefficients.
That's right. And critically, your sources highlight that only the unbound drug in the plasma,
the free drug,
is available to leave the bloodstream and actually act on targets in tissues.
So plasma protein binding is key for that unbound concentration. Drugs binding to albumin, etc.
Exactly. And it's not simple. Models need to account for binding to multiple proteins. PPB can be predicted computationally using QSRR or ML models.
A major barrier for brain drugs is the blood-brain barrier, the BBB.
Huge challenge. Getting drugs across this highly selective barrier, it's tough.
And predicting BBB penetration computationally is complex. Models predict log BB.
Often, yes. The ratio of drug in brain versus blood. But, as the sources note, that's indirect. It doesn't tell you the unbound concentration in the brain, which is what really matters.
Factors like TPSA, polar surface area, and log-log D are used in simpler equations.
They are. But the BBB itself is complex, tight junctions, transporters pushing drugs out.
Modeling specific transporters at the BBB is key then, like the choline transporter for getting drugs in.
Yeah, using techniques like homology modeling and docking to understand those molecular interactions.
For overall distribution modeling, PBPK models are the go-to again.
Definitely. Because they have organ compartments, they can use mechanistic equations or ML predictions to estimate K-through how the drug partitions into each tissue based on blood flow, composition, drug properties.
So software like PKSim and SimSip integrate drug properties and physiology to simulate whole body distribution.
Right. The examples show PKSim predicting lamotrigine levels well in a specific patient group, helping with dosage.
And SimSip models validated against clinical data for drugs like ondansetron.
Exactly, incorporating those predicted K-free values.
Okay, as the drug is distributing, it undergoes metabolism, the body changing the drug chemically.
Often to make it more water-soluble for excretion, yeah. The liver's the main engine, but other tissues contribute.
Driven by enzymes like the cytochrome P450s, the CYPs.
That family is crucial. Predicting how a drug will be metabolized and by which enzymes is vital. And it's not just simple rules like Lipinski's anymore. Newer descriptors are needed.
Understanding interactions with metabolic enzyme substrate or inhibitor is key. Pharmacophore modeling helps here.
It does. Pharmacophore modeling identifies the essential 3D features a drug needs to interact with an enzyme's binding site, like a hydrogen bond spot here, a greasy patch there.
Software can generate these models.
Yes, and if the enzyme's 3D structure isn't known.
Homology modeling, building a structure based on similar known ones.
Right, using tools like Swiss model based on the protein sequence.
Once you have a structure, molecular docking simulates how the drug fits in.
Yes. Software like Autodoc Vena simulates the binding, predicting orientation and strength, which relates to how well it might be metabolized.
And QSRME models predict metabolic outcomes directly from structure.
They do, predicting which CYP metabolizes it or estimating liver clearance rates. Lots of software packages exist for this.
The final stage, excretion. Getting the drug and its metabolites out. Kidneys, liver into bile.
Primarily, yes. Plasma clearance is the overall measure. Renal excretion involves filtering, active pumping by transporters, and sometimes reabsorption. Biliary excretion uses transporters in liver cells to move drugs into bile.
PBPK models are invaluable here too, simulating kidney and liver physiology.
Absolutely, including transporter activity. They can simulate clearance in healthy people and those with organ issues by adjusting parameters like GFR or transporter levels.
QSPKR models predict excretion aspects too, like fraction excreted unchanged.
Yeah, or likelihood of active transport in the kidney. ML models are also trained on clearance and transporter interaction data.
Transporters really are central to so much of this ADME process.
They are absolutely essential. Predicting drug transporter interactions is critical for understanding absorption, distribution, metabolism linkage, and especially excretion.
And predicting drug interactions, right? One drug blocking a transporter needed by another.
Exactly. That can lead to dangerously high drug levels. The sources detail modeling for key transporters like PGP, BCRP, OCTs, OATPs.
Using QRL to predict substrates inhibitors.
Often with high accuracy, yeah. And if the structure is known, structure-based docking gives more detailed insights. Recent cryo-EM structures are helping a lot here.
Accessing data is fundamental for all these models. Databases like PubChem, DrugBank.
And specialized transporter databases. They provide the structures, properties, activity data needed to build and test the models.
So the general workflow often involves mining databases, calculating descriptors, generating pharmacophores, maybe homology modeling, then docking.
That's a common sequence, yes.
Yeah.
Depending on the specific question and available data.
It's clear this field is incredibly powerful, but it's not without its hurdles. What are the current challenges?
A big one your sources mention is data quality and availability. Good models need good data.
And data can be variable or incomplete.
Especially for less common transporters or diverse patient groups, yeah. That hinders model development and validation.
Model complexity versus interpretability.
Right. As models get more complex, understanding why they make a prediction becomes harder, but it's crucial, especially for regulatory acceptance. Transparency is key.
AML faces challenges with explainability and generalizability.
Yes, and gaining full regulatory trust. Hybrid models combining AI's pattern finding with mechanistic understanding seem promising.
Modeling less studied transporters and complex interactions is still tough.
It is. Limited structural data and interspecies differences make extrapolation tricky.
Looking ahead, what's next? Where's this field going?
Well, continued efforts to build and share high-quality, comprehensive databases are essential.
More structures from cryo-EM.
Definitely. Better enzyme and transporter structures will fuel more accurate structure-based modeling.
Integrating omics data, genomics, proteomics.
Yes. Incorporating that will allow for even more personalized systems-level models, predicting behavior in specific individuals based on their unique biology.
Explainable AI and digital twins. Virtual patient models.
Exciting directions, yes. Advancing precision medicine and regulatory science.
And standardizing models for regulatory use.
Crucial for practical impact. Harmonizing validation, reporting.
Yeah.
So regulators worldwide can trust and use these models consistently.
Plus education, right? Training scientists in modeling and data science.
Absolutely vital for future progress. So we've really traced that journey of a drug molecule through the body absorption, distribution, metabolism, excretion, and seen how computational modeling gives us these powerful tools to understand and predict each stage.
It's this complex interplay, isn't it, between the drug's chemistry and the body's biology.
And these computational approaches, well, they essentially give us X-ray vision into that process. It's not a black box anymore.
It's clear that these in silico methods, PBPK, docking, AI, all of it, aren't just supplementing experiments anymore. They're enabling a fundamentally more rational, more efficient way to design drugs.
By pulling together all this diverse data chemistry, biology, physiology, and using sophisticated algorithms, researchers are making much more robust predictions much earlier in the process.
Which is vital for reducing that huge experimental burden.
Absolutely. And accelerating the pace of discovery. And ultimately, supporting personalized medicine by helping predict how a drug might perform in different individuals or populations.
What started, as you said, with just finding a molecule that hit a target has evolved into this incredibly sophisticated blend of chemistry, biology, and computation.
Predicting the whole story of the drug's journey.
Ah.
And as data gets better, as we unlock more protein structures, and as AI becomes more transparent and explainable, the accuracy and predictive power of these models will only increase. It's really pushing the boundaries of pharmaceutical research.
So you've had this deep dive into your stack of sources. The big takeaway isn't just that computers are used in drug development, but how they're being used to model, predict, and optimize that fundamental process of getting a drug into your system to the right place, doing its job, and then safely removed.
That whole ADMET picture.
It really makes you pause and think about the sheer amount of complex, invisible engineering that goes into designing a safe and effective pill, doesn't it?
It certainly does. And seeing the push to standardize these models for regulatory use shows just how integral they've become. This isn't just academic anymore.
No, it's changing how medicines reach patients.
Exactly.
Something significant to chew on. And hopefully this deep dive helps you feel significantly more well-informed about this critical high-tech area of pharmaceutical science.